Hello, microbiologists. So let's talk about how microbes grow, how they grow in the wild as biofilms, and how they grow in the lab on media. So in the wild, microbes are going to grow as communities. They're very seldom are they going to just be all on their own. No bacteria is an island. They often are going to be kind of covered with a slime. That slime is going to be a mix of polysaccharides, DNA, and proteins. We're going to skip talking about hydrogels. Bacteria within a biofilm are going to be able to communicate with each other. And the way that they communicate is called quorum sensing. So each bacteria will secrete a little chemical signal, and together they can use those signals to kind of coordinate their activity. They can tell how many bacteria are there, so they have enough bacteria to, do something together. They can coordinate what they're doing. They can group into communities. They're just able to use those little molecules, chemicals that they emit to signal each other. One example would be if you've ever seen those fish, I forget what they're called, those fish that live deep in the ocean, and they have a little light organ over their head that can light up. It's bacteria within that light organ that's creating the light. And the bacteria are only going to, it takes a lot of energy for them to emit light. They're only going to do that if they know there is a lot of bacteria there. And so it's through quorum sensing that they're able to tell that there is enough bacteria to create a noticeable level of light. So they all group together and turn on their light together. Biofilms could be made of just one single species. They also could be a diverse group. And they're going to be attached to some sort of surface. So if you've woken up in the morning and your tongue kind of slips over your teeth and they're a little bit like, there's something on there, that's a biofilm. If you've ever gone into your shower and your shower curtain has just a little bit of a slimy black layer on it, that's a biofilm. If you've ever stepped into a pond and you stepped on a rock and it's a little bit slimy, that also is a biofilm. The reason bacteria grow in a biofilm is because there's lots of benefits for them. One, allows them to share nutrients. So if one of them finds a lot of nutrient, they share it with the group. They stay better fed. And by having that increase in energy available to all of the cells, they're all growing. They're also sheltered from the environment. So if you're looking at this, as groups, if there's something in the environment that's not so great, maybe a little bit on the surface, but a lot of that slime around it is going to really protect the cells. And the cells within the middle of it definitely are protected. They're also protected from desiccation. That means drying out. They're protected from antibiotics. So the antibiotics come in and, there's like a sugary layer, but there's not really any signs of bacteria. They're sheltered from the immune system. The bacteria growing together are able to transfer their genetic information. If you remember talking about a sex pilus in chapter 4, they're able to connect to each other and just transfer plasmids to each other. They're also able to increase resistance to microbicides, both because any microbicide around it is not able to get into it, but also because any bacteria in here that has a little bit of resistance is able to share that resistance with all the other ones. Biofilms can be found in the digestive system. They do grow along your mucous membranes where they protect you, and they are very important there. However, they also can clog pipes. Every now and then you might hear a news story of, this pipe was just cleared out of this. Look at this huge, gross sludge we just pulled out of it that's made of biofilms. Medically, a lot of infections can be linked back to biofilms. So catheters, heart valves, contact lenses, dental caries, or that means cavities, those can all be linked to biofilms, bacteria that are growing on that catheter or that valve, that implant, a bacteria film growing on the contact lenses or on your tooth. And once they're growing there, they're very difficult to treat because if you remember, they're more resistant to microbicides. So it's a lot easier to use aseptic technique and reduce the risk of an infection. But eventually, if you have a biofilm infection, you're going to pump up that medicine to kill it. So why are some reasons pathogens would grow as a biofilm? How is that advantageous to the pathogen? Then let's talk about how we grow bacteria in the lab. In the lab, we want to grow pure cultures, or that means a culture made of only one microbe. So some different terms when we're referring to growing bacteria: a colony, a culture, and a pure culture. I will let you read these terms because I don't think you want me to just read it to you. Culture media is the nutrient material that you are growing your microbes on. So we have used TSA a lot as our nutrient, as our culture media. It has all the nutrients a microbe needs. The inoculum is the microbes that you put on the media. So when we did our thumbprint lab, your thumbprint was the inoculum. When you swabbed a surface, that surface is the inoculum. If you are taking a sample of bacteria from one tube and putting it onto the plate, that first tube is your inoculum. And then streak plate method. This is something we do in lab 4 and in lab 6, where you take a sample of your bacteria and you just spread it out over this first section. Then you flame your loop, so there's nothing left on your loop. And in your second smear, all you're doing is taking this first section and thinning it out. Then you flame your loop, so there's nothing left on your loop. And you do a third swab, where you're just thinning out that second section. So you can see that first section, you've got tons of microbes growing there. It's going to be very hard to pick out one individual colony to get a pure culture. But then you've thinned it out, and when you thin it out this second swab, you're starting to get some individual colonies. And then the third swab has even more individual colonies. And those individual colonies, you can assume each one of them grew from one bacteria. They each contain clones. And you can take one of those colonies and use it to make a pure culture, or a culture containing only one species or strain of bacteria. There are some different types of culture media that we'll use in lab. In a chemically defined media, you know exactly what is in that media. You know I have exactly this many micrograms of sodium, this much of each amino acid. I have this much glucose. I have this much of DNA. I know exactly what's in it. In a complex media, I don't know exactly what's in it, but I know it's got everything that my microbe needs to grow. Some types of complex media would involve taking like kelp and blending it. And you don't know exactly how much sugar is in that kelp. You don't know exactly how much amino acid is in there, but you know it's got everything to sustain life. Some complex media involves sheep blood. You don't know exactly how much, iron and oxygen is in that sheep blood, but you know it's got everything you need. Then reducing media. This is a definition that trips up everyone every year. So I don't know what the secret is here, but pay attention to reducing media. Reducing media reduces oxygen. It has nothing to do with redox that we talked about in chapter 5. Reducing media reduces oxygen that's available there. And when it reduces the oxygen, it removes the oxygen, reducing media will let you grow anaerobes. If you think about when we've grown stuff in the lab, it's just open on a plate. You can't grow anaerobic bacteria that way. So we need reducing media to grow anaerobic bacteria. Selective media and differential media. This is stuff that we'll do in lab when we start working with MSA or with EMB culture. These are selective and differential. Selective media will select what bacteria it wants to grow and won't let other stuff grow. So selective media, you'll only have certain types of bacteria growing. So maybe it's a media that has a lot of salt in it that only grows halotolerant bacteria. Maybe it is a bacteria media that only grows gram-negative bacteria. Those are selective media. Differential media, see that root different in there? Differential media makes things look different. So this would be a type of differential media. Staph epidermidis grows, the media stays red. Staphylococcus aureus grows, and the media turns yellow. So the media changes color because it's differential. Some types of differential media, the whole media changes color. That's what you see here. The whole media is turned color. In some types of differential media, only the bacteria would turn color. So you would only see, specific dots that are purple or green or something, depending on what type of differential media it is. Then once you've grown it, you're going to preserve it. The way we preserve bacteria in the lab is usually these 3 different ways. One is refrigeration. Refrigeration is great for short term. It's nice and cool, so your bacteria isn't going to grow. It will eventually dehydrate your bacteria and kill it. But refrigeration is good to keep things for a week or a couple of weeks. In lab, you are growing things. We stick them in the incubator to grow them. And then from the incubator, they go back to the fridge until your next lab because we don't want them to just keep growing all week. So refrigeration is used for short term. If you're going to store things for longer term, months or probably months to years scale, you can deep freeze it. We take your pure culture, you put it in a specific suspending liquid, you freeze it, and you can keep it at temperatures up to 95 degrees below zero. So this stores things for longer term. Lyophilization refers to freeze drying. So it dries it, it removes the water, it vacuums out the water, and it also freezes them at freezing temperatures down to 72 below zero. And it's going to be, because it's dehydrated, it'll be kind of powdery, and you can keep that for years. When you're ready to use it, you just mix the water back in to rehydrate it, and you can start growing your bacteria again. So here's a chemically defined media. You know exactly how much glucose you have used. You know exactly how much of each of these salts. So you can say, I know exactly how much phosphate, how much chlorine, how much magnesium I have. I know exactly how much of each amino acid I've added. I know exactly how much of each nucleotide I've added, how much of each vitamin. Everything is defined. It's a chemically defined media. Here is a jar where you can grow anaerobes. Do you remember what kind of media that's called? In this jar, I seal my microbes in here. So here are my plates that are growing my microbes. And this is going to remove the oxygen. It's going to react with oxygen in the air to remove it. Do you remember? That is reducing media. Please keep that definition straight in your head. Okay. Then let's end with talking about biosafety levels. So we can categorize bacteria based on how dangerous they are. At a biosafety level 1, there's no special precautions. This would be stuff like, the bacteria that makes yogurt. Absolutely not dangerous at all. At biosafety level 2, these are things that could cause an infection, but they're not hugely communicable. They're not hugely dangerous if you did get sick, and they're treatable. So at a BSL2, you'll wear a lab coat, gloves, and eye protection. This is where we stay, level 1 and 2. Most of the things we deal with in lab are level 1, but some of the things are level 2. So we do wear a lab coat, gloves, and eye protection. At biosafety level 3, you need to use a biosafety cabinet. We do have these cabinets or hoods within our lab, and you can ask me or your lab instructor if you have someone else to show you what these hoods are like. For biosafety level 3, these are things that are going to be starting to get a little bit more transmissible through the air. So these hoods will prevent them from traveling through the air. They have negative air pressure to draw the bacteria in. But these are still things that are treatable. At a biosafety level 4, this would be like hot zone. This is what's going on in this screen. They are, well covered. This would be something like Ebola, things that are dangerous, not necessarily treatable, not necessarily easy to contain, things that you under no conditions want to be spread out. So you have this fume hood you're dealing with that any air filters are going to have, twice filtered through HEPA filters. They are breathing in positive pressure air, which keeps the microbes out. They have to go through decontamination. There is secret codes to get into the lab. This lab is, under lockdown. People can't get in it. Level 4 is like the terrifying level. Okay. So in theory, not necessarily a fun, enjoyable vacation survival, but could humans exist on a chemically defined media under laboratory conditions? If, some alien invaders came and put us in little people zoos and fed us chemically defined media, could we survive like that? Could they say, here is how much sugar, how much lipid, how much protein, how much nucleotides you need, and you're eating that? Could you survive? Maybe not happily, but could you exist? What BSL level is our laboratory? What kind of safety techniques does that mean we need to use? When you are growing bacteria on a Petri plate, what could prevent a colony from growing infinitely to just infinite size? Or at least what could prevent it from growing to just fill the Petri plate? And could a pure culture of bacteria be obtained by the streak plate method if there were only one microbe in a suspension of billions? Okay, these are a little bit more thought questions, not necessarily that just straight up go back and see the answer and copy it out from the past slides. So we're at a point now where we're thinking a little bit more about what we've been talking about. I hope this was a little bit more interesting to you. It's pretty relevant to what we're doing in lab. If you have questions, let me know, and I'll see you for our next video. Bye.